Hi there, everyone. My name is Jacob Nikolcha. I am a second-year medical student at the Perelman School of Medicine at the University of Pennsylvania. I want to take a moment to talk about flash radiotherapy, and specifically flash radiotherapy for the treatment of glioblastoma. As a medical student, I am very interested in radiation oncology. Currently, I am on my neurosurgery rotation, and I am doing this presentation to sort of intersect the two and enhance my learning and share something about a topic that's really interesting with me. So a few disclosures first. The work that I'm describing in this presentation is not my own, and I'm not affiliated with these investigations in any way. This presentation is an exercise for me as a student, and it's largely a summary of work that's been done by Montay-Gruel and Acharya and colleagues. With that said, let's get into it. So what is this so-called flash effect? Let's unpack that a little bit. Firstly, I think we need to understand a little bit about conventional radiation before we can understand flash. So most radiation therapies are delivered in the range of 0.07 to 0.1 gray per second. The flash, on the other hand, is given at really, really high dose rates. And it's this paradoxical finding that actually, when you deliver radiation at these really high rates, like 100,000 to 1,000,000 gray per second, so giving whole treatments in a fraction of a second, in certain cases, it's actually less toxic to healthy tissue, but equally toxic to tumor as regular radiation. And that's really a paradox because you think about radiation in the public perception, you think radiation is dangerous. A lot of radiation all at once must be super dangerous. And in fact, it turns out that in certain scenarios, in certain cases, in certain malignancies, it's actually less toxic to go with these high dose rate methods. And that is a little bit of a mystery, and the mechanism there is not fully understood. But the potential for clinical benefit is something that we're currently in the process of trying to learn about and trying to exploit. Moreover, most current conventional radiation therapies are split into many smaller doses called fractions. And these fractions build towards a target dose across days to weeks. So, let's say you want a total dose of 12 gray, you might get 2 gray today, 2 gray tomorrow, 2 gray the day after that, until you get 6 treatments, and then you're up to your total dose of 12 gray. Flash, on the other hand, people take a different approach. Remember, you can give a whole radiation treatment in less than a second. And so for that reason, flash is often delivered in fewer fractions, or that's at least the theoretical idea behind it. You can give it in fewer fractions or potentially a single treatment. These things are all kind of being worked out and explored currently. So let's talk a little bit more about why flash is useful. Again, less toxic to healthy tissue in the right situations, but equally toxic to a tumor. Potentially less radiation-induced neurocognitive effects. If we're thinking about cancers of the CNS, like glioblastoma, that's really important. So decreased neural inflammation, decreased neuronal degradation. These are all really, really important things in the setting of a CNS malignancy. And there's also this possibility for dose escalation. So radiation therapies are often limited by the side effects that they cause to healthy tissue. And that's true in glioblastoma models. That's true in a lot of different ones. And so if you're able to reduce the effect on healthy tissue, you can potentially actually raise the dose to get better targeting of malignant cells. And then again, like I mentioned before, this potential for shorter, fewer treatments. And that's just easier on patients. That's easier on radiation therapists. It's more convenient. And it's definitely a potential benefit of this type of strategy. So how has flash been applied to GBM? First of all, the good news. mice GBM models that are treated with flash seem to retain better cognitive function than those treated with conventional radiation. The bad news is that mice are not human beings. And I'm on my neurosurgery rotation, and everyone I talk to about flash radiation is quick to bring that up. And it's true. It's not the same thing. And then it's also tricky because the parameters of dosing and fractionation really matter. The flash effect kind of has to be teased out a little bit. And people are still trying to work together and figure out what are these parameters. And we'll discuss that a little bit more going forward. But more good news is that GBM mouse models have actually served as a platform for characterizing some of these parameters. And again, I'll show some of that data. So here's some early results from a GBM mouse model study of flash radiation. And I think I'm just going to talk through this a little bit so that I can show everyone the trend that I'm talking about and really try to hone in on what flash is. So in these studies, on the y-axis here, this is basically a measure of mice like cognitive function and memory. And it's a standardized experimental method for measuring like basic mouse cognition. And here in the blue, these are mice that these are all mice that have glioblastoma. The ones in the black are not treated with any radiation therapy. The ones in the blue are treated with conventional radiation, and the ones in orange are treated with flash. And what you can see here is that at a dose of 10 gray, given all at once in one fraction, mice that are treated with flash don't have any change in neurocognitive function compared to controls, whereas the ones treated with conventional radiation see a sharp decline. And so that's the flash effect right there. So with this, the authors decided, let's try to bump up the dose and see if we still see the flash effect. So they bumped up to 14 gray, and unfortunately, they didn't see it. It's just a 4 gray bump in the dose, and suddenly the flash effect isn't there anymore. That's a little curious. So the next thing they did was, okay, let's split 14 gray into 2 fractions, so 2 7 gray treatments. And lo and behold, when they did that, the flash effect comes back. That's really interesting there. You can start to see that if you go a little bit too fast all at once, no more flash. You split it up, and suddenly flashes back. And so we'll think about this a little bit more going forward, but keep this thought in mind. So the next thing they decided to do was, okay, let's break up the fraction even more. Let's do a total dose of 14 gray, but given in 4 fractions. And boom, flash is gone again. So that's curious. You give it in 2 fractions, flash is there. You give it in 4 fractions, flash is gone. What could that mean? Then they realized that, like here in this experiment, 10 gray showed the flash effect. So they decided, okay, let's give a couple fractions of 10 gray and go up to a total dose of 30. And boom, flash is there. So that's really interesting because you start to see that once you achieve the flash effect and you find a dose that works, it looks like you can stack that across multiple fractions. So they have total dose here of 30 gray, but no change in cognitive function. That's really incredible. And that is the Goldilocks zone there. And that is the potential of flash. And so a picture is beginning to take shape here, which is that flash works, but dose and fractionation really matter. So again, let's recap this a bit. At 10 gray in 1 fraction, you get flash. Just a little bump up in dose, that's too fast. But then you split it up a little bit, suddenly flash is back. Split it up too much, no more flash. But then if you take one of these doses like 10 gray and you keep giving 10 gray fractions, you keep that flash effect across the entire dose spectrum. So think about it, like treating to 14 gray, and you get serious neurocognitive declines. But here with the flash effect, you get up to 30 gray, which is double, more than double, and no change in neurocognitive function at all. And so again, it is that Goldilocks zone, and that is what people are chasing with this type of research. This is just explaining the same thing, but in a more visual form, explaining some of the doses that show this neurocognitive sparing and the ones that don't. And I think I walked through this trend a little bit, but really what emerges here is that the flash effect is in a sweet spot. And when you hit that sweet spot, these neuroprotective effects persist even when you start building towards a higher cumulative dose. But of course, the limitations are mice are not human. And also these studies use whole brain irradiation, meaning that the radiation field that they're using it encompasses the entire brain. Now, in humans, this would not be the approach we would take. We have much more precise methods for targeting the actual radiation beam to be more shaped to the actual volume of the tumor while avoiding the healthy surrounding tissues. And so I think that is one very big difference in the way that we would use flash in the real world. And it is definitely a limitation of this study. And finally, measuring cognitive function is not easy. It's not easy in a human, less easy in a mouse. And it's even less easy when the thing you're trying to measure, the treatment and the disease, are both affecting cognition. And so they confound each other. And that is an inherent limitation of studying cognitive effects in a GBM radiation model. Now, I will say this same group has a study where they perform radiation on mice that have on mice brains that have no malignancy, and they still show that this flash effect persists even in mice with no tumors. But at least within the context of this study, it is one thing to keep in mind. So the major takeaway here is that flash is real. It's becoming more accessible, at least in sort of a research sense, meaning that people are starting to figure out the doses that actually achieve this effect. And it has the potential to be a part of a changing approach that radiation oncologists are taking with dose and fractionation studies, which is this trend towards what is called hypofractionation, meaning an approach that kind of favors a smaller number of higher dose fractions. And in certain cases, in certain tumors, in certain malignancies, you can achieve this sweet spot of treatment efficacy, toxicity, and also just convenience for patients. And flash is kind of like the most extreme example of this kind of trend within the field. So what are the remaining needs? We need to better characterize the parameters that are achieving flash, and I'll go into that a little bit. We need to better understand the mechanism of flash. Again, very paradoxical that you can shoot these radiation beams so fast that you give a whole treatment in less than a second, and somehow it's less toxic. It's really, really kind of backwards. And there are a few ideas of how this works, and we'll go into some of those as well. Then flash needs to be brought into the clinical practice. I think the practice of radiation oncology is full of times when maybe we didn't fully understand the mechanism or fully understand the parameters, but once we started seeing a clinical benefit empirically, it became a part of practice. And I think flash could be one of those things as well. And then I guess with flash being as trendy as it is within the field of rad-onc, and with all trendy things in science, really it ends up trickling down to finding which patients actually benefit the most and which subset are appropriate candidates for this type of therapy. And so that is another next step that needs to be taken. So let's talk a little bit more about some of these parameters that achieve flash, and also a little bit about flash from a mechanistic standpoint. So parameters that achieve flash, this is data that is a collection of different parameters used in different studies and sorted by which ones achieve flash and which ones don't. I want to point out that work is being done at our institution in this regard. And what is being found is that instantaneous dose rate, pulse frequency, total dose, fraction size, number of fractions, like all of these things all matter. And this is like some really nerdy like medical physics stuff. And I gotta admit, like I do not fully understand this stuff. But it turns out that if you want to tease out this flash effect, these are all things that need to be thought about and set up appropriately. And so that leaves flash as really an optimization problem where a lot of variables are contributing, and it's up to us to sort of tease those things out. Onto the proposed, excuse me, onto the proposed mechanism of flash. Current data is suggesting that flash works by generating fewer reactive oxygen species, and especially within healthy tissues. And so this is something that has been measured by the same group that we've been talking about. And also they've done experiments where if they double a mouse's oxygen tension, it turns out that it reverses the flash effect. Like flash doesn't exist when the brains of these mice are loaded with a lot of oxygen. And so it further lends some support to this idea that reactive oxygen species is playing a role. There's a possibility that the immune system is playing a role. However, I don't think it is the dominant effect here, because a lot of these studies have actually been done in completely immunodeficient mice and still showed the flash effect. And yes, recap, these data are indeed suggesting that reactive oxygen species insult, and a difference between that in tumor cells versus healthy cells is part of the process that is mediating the flash effect. So the future holds a few things for flash radiotherapy. Like I mentioned, this trend towards a lower number of higher dose fractions, and a lot of that being driven by empiric clinical benefit. And also just better define the parameters that achieve flash, and allow, and more institutions also need to adopt the technology necessary to support this type of therapy. Having linear accelerators that are capable of achieving the dose parameters and really high dose rate necessary for flash is a challenge, and that is a technological and logistical challenge. And so more and more centers are looking into this type of thing. And so before I go, I just want to mention some interesting work that's being done here at Penn, and is just for fun, but actually really important. Maple is a really adorable and brave service dog who was actually recently treated with flash here at Penn. And so the vet school here is actually very interested in flash as well, and is starting to expand into animal models with dogs. And so dogs like Maple are helping us learn how to better treat both people and pets alike. And I think that's really super adorable. And if you want to read more about it, I've included the link there. So with that, that is all I have for tonight. And if anybody has any questions, my email is up on the first slide. If anyone just wants to chat about radiation or neuro-onc or any of these things, I'm more than happy. Or if there's something in this presentation that's wrong and somebody wants to shoot me a message, I'm more than happy to look into these things. I'm a student. I will always be a student. I'm a lifelong learner. So I am more than happy to learn more. So with that, take care, everyone.